we go over the Lac Operon. Now the first thing that we want to get to understand is what the Lac Operon is and also its function. How does it carry out its function? So those are the works we want to understand by the end of the video. And so Lac Operon is a piece of bacterial DNA So this is a piece of bacterial DNA that has got some genes which are able to break down lactose. So that is what is referred to as a Lac Operon, just a piece of bacterial DNA that is responsible for the breaking down of lactose. So it has got some genes, and those genes can produce what is necessary for the breaking down of lactose. Well, an example of a bacteria that we are going to consider for us to understand this process is E. coli bacteria. Now, what happens, mostly bacteria is one organism that is found in the body of even human being, in the stomach. And so they are necessary actually. So they are going to help us by breaking down the food that we eat. At the same time, when they break down the food that we eat, they are also going to feed on the same food. And so that is mutualism, where we gain from them and they also gain from us because they find an environment in us for a stay. And so the E. coli does not use lactose, mostly. So that means if we eat food that contains lactose, that is, if we take milk, most of the times the E. coli bacteria does not use the lactose. And what is the reason behind that? That is because when we want to use lactose, when the bacteria has to use lactose, it has to be broken down. So the lactose has to be broken down into glucose and galactose. Well, this process doesn't just happen. It needs energy. And so the reason as to why the lactose has to feed is to gain energy. Now at the same time, for it to have energy, it has to use energy again. That is why it is very difficult for the bacteria to just go on direct and use lactose. And so it would rather, if there is lactose in the body and also glucose, lactose shall never be used until all the glucose has been used up. So it is not possible to start using lactose when glucose is there. Not until all the glucose has been used up and there is nothing that is there to provide us with energy. That's when lactose will be broken down by the E. coli bacteria. And so that's when now the Lac Operon comes in play. So this piece of DNA comes into play. Well, let's consider the structure of this Lac Operon. Now, generally speaking, we need to get to realize that any kind of gene, or let's say just DNA is divided into too many groups. So when it comes, we are going to classify DNA into groups. There is what is called the structural gene and also the regulatory gene. So this one which is in front here, which I've put in green, that is going to be the regulatory gene or the regulatory DNA. And then the one at the end, this is going to be now the structural gene, or simply a sequence, which is responsible for structures. And so if you have got a gene here which is saying air has to be produced, or you need to produce an enzyme that has to break down lactose, before you actually produce that enzyme that has to break down lactose, it needs to receive a command from the regulatory gene. So the regulatory DNA contains two portions. We have got first the regulatory gene, let's say the many regulatory gene, and also there is another part that comes there which is just the regulatory sequence. Alright, so this is all the regulatory DNA, which contains regulatory sequence and also the main regulatory gene. Well, what do these do then when in reference to what we are discussing? Now, before the gene, so what we are having is, let's say we have got a gene A and then another gene B. So these are responsible for something that has to be produced. In this case, because we're talking about the Lac Operon and the breaking down of lactose, let's say these genes are responsible for the production of enzymes which break down lactose, because lactose will not just be broken down, it has to receive command, and before that command comes, these have to produce, because these are the ones which contain instructions that lactose have to be broken. So the information which they contain have to be converted now into the right enzyme. For example, A might say synthesize an enzyme to catalyze the breakdown of lactose. So that means if A hasn't produced that instruction, if A hasn't produced that what we call gene product, okay, what comes out from a gene, that enzyme which is supposed to come out from the instruction of this, then you're not going to have the breakdown of lactose. Alright, so before the genes which code for proteins A and B, we have what we call the regulatory DNA, as we can see, and the regulatory DNA is separated into two. We have got the regulatory sequence and the main regulatory gene. So the regulatory sequence just lies a few hundred bases from behind the coding gene. So these are the two coding genes. These are the ones which are going to code for some gene product, such as an enzyme. So this sequence just lies a few hundred base pairs behind. Well, what is the function then? So this point, the regulatory sequence, is a point of attachment of RNA polymerase. So this is where RNA polymerase is supposed to attach. And you know, this is the enzyme that has to synthesize RNA from DNA. And so RNA polymerase is going to attach to this portion and it starts moving like this. When it starts to move, that's when it is going to be able to read this being converted into now the right gene product. The gene product which can be responsible for the formation of any enzyme that can break down our lactose. Alright, so we are saying this is a point of attachment of RNA polymerase. RNA polymerase is an enzyme that is responsible for copying of genes which are on A and B onto mRNA during the process of transcription. So the information that is in this, because directly, even if this A contains information to say we need to produce an enzyme to break down lactose, it is not DNA which carries out that information. The job of DNA is only to store the information, and so what is going to carry out that is mRNA. And so this has to be first converted into mRNA. That is why RNA polymerase has to attach to the regulatory sequence and then be able to convert now the information found on this gene into information on an mRNA molecule. Well, we also have the main regulatory gene just behind the regulatory sequence. So the main regulatory gene, this is responsible for the synthesis of a protein that is known as the regulatory protein. So it synthesizes a protein, regulatory protein. Well, what is the work of that regulatory protein? And so the regulatory protein goes on and controls the binding of the enzyme RNA polymerase to the regulatory sequence. So that is the function. This one controls the production or synthesizes this enzyme which is called the regulatory protein. and then this protein is going to be responsible for the attachment of RNA polymerase to this region before the RNA polymerase starts to move on here. So that is it. Well, when that happens, when this enzyme has been synthesized, and then it controls the binding of RNA polymerase to this sequence, that now is going to be able to either decrease or increase the rate at which transcription happens, depending with what we want. So if we don't want the process to happen, and if we want the process to happen, and that depends with the need. Alright, and so the regulatory sequence, just this portion we are able to see here, is not that simple. It is also divided into different parts which are able now to control this portion, because this regulatory sequence is the one where the RNA polymerase has to lie. And so the RNA polymerase doesn't just start there, immediately it comes there and then it starts to move. It's a control system. The regulatory sequence is a control system for RNA polymerase. And so the regulatory sequence also is further divided into different parts which are necessary for the conversion of our lactose, okay, into glucose for us to gain energy. Well, now because of this, let's go now in detail and understand a Lac Operon. So this is the basic structure of any gene. Now from here we understand the structure of Lac Operon. So let's say this is a gene. We have got these parts. We said we have got the structural gene. So these are the structural genes, the three of them. This is for a Lac Operon. So I'm going to put them like that to show that they are one. They are all structural genes. And then we have got behind here, this is where now we have got the regulatory DNA. Let me put it in different color then. So this is what is responsible for the regulation, for the controlling of structural genes. So this is the regulatory portion. Well, what you need to get to understand is these are the two basic structures of a gene. A gene basically has got only the regulatory, which controls the structural. And so structural genes, these are responsible for the production of gene products such as proteins. And then before these are able to produce what we are calling as gene products, they have to be controlled by regulatory genes. So the regulatory genes, or the regulatory portion, controls the structural genes. And we need to get to know that in a cell, actually the structural genes is just about 2%. The other 98% is just there for the controlling. So what is responsible for forming gene products? Just 2%, and then the rest is just to control the formation. Now, a Lac Operon, we've already said what it is. So a piece of DNA from a bacteria that is responsible for the breaking down of lactose. Okay, now it does that by coding or producing some enzymes which are necessary for the breakdown. Just as we can see, the same genes we are able to see here are going to produce some enzymes which are necessary to break down the lactose, because there is a bond between glucose and galactose which makes this to become lactose. So that bond has to be broken. So that is really what happens. And so let's go on. So the Lac Operon can be again broadly divided into the parts that we've seen here. So where this is called Lac Z, this is Lac Y, and this is Lac A. And each of them have got their own function there. Okay, we have got another part here. Let me just include it. We are going to understand it later. And so The Lac Operon is divided also into these two portions, the structural genes and the regulatory portion, as we can see. Now the structural genes code for enzymes, while the regulatory portions control the structural genes, as we've already seen. These are going to code for enzymes which are able to break down the lactose, and then these are going to be controlling these which are here. Well, the regulatory portion, this portion we are able to see, is actually what switches on. The main function is to either switches on or off the structural genes. Because we are saying it controls, the same word to control means to switch on or off. So there are three important structural genes in the Lac Operon, as we can see: the Lac Z, the Lac Y, and the Lac A. Now, they are all responsible for something. So we have got Lac Z, Lac Y, and Lac A there. Alright, so what does each of these do? Well, starting with Lac Z. Lac Z codes for an enzyme which is called beta-galactosidase. So this is beta-galactosidase. Now, what is the main aim of us producing this enzyme called beta-galactosidase? Well, beta-galactosidase breaks down the bond between glucose and galactose. So this is the enzyme that is responsible for the breaking down of the bond between glucose and also galactose, because we know that this is what forms the lactose. So actually, lactose is going to be broken down into glucose and the galactose. Now they are going to be individually. The enzyme responsible for that is beta-galactosidase, which is produced by Lac Z. So just the first one, which is here. Alright. So another one which we have here is Lac Y. Well, Lac Y also codes for an enzyme which is called beta-galactoside. So this is galactosidase, this is galactoside. Now, what is the function of beta-galactoside? So this is actually beta-galactoside permease. Well, even from the word we can hear Lac Y codes for the enzyme called beta-galactoside permease. Just the word permease coming from the word permeable. So this one fits in the cell membrane of the bacteria and allows more lactose to be pumped into the cell. So that is a function, permease, permeability. It is going to fit into the cell membrane of the bacteria. So if we have got this as our bacteria, it's going to fit past there. When this beta-galactoside permease passes into the bacteria, it's going to force a lot of lactose to be deposited into the bacteria, thereby making sure that beta-galactosidase breaks a lot of it, and we are going to have a lot of glucose at the end of it all. So that is the function of beta-galactoside permease, to be able to pass through the cell membrane and deposit more of the lactose, which can be broken more and form a lot of glucose. Well, Lac A is responsible for the enzyme beta-galactoside, so it codes this is also beta-galactoside. Okay, that is transacetylase. Well, for this one, it doesn't have much important function for our discussion, like A. So the main point of our discussion, it doesn't have any useful function for this one on the Lac Operon for now on this discussion. So the main ones is beta-galactosidase and beta-galactoside permease. permeates to penetrate the cell membrane of the bacteria and force more lactose to enter for breaking down. Galactosidase is the breaking down of our what? Our lactose into glucose and galactose. Well, these are the parts. Now let's go on and see, because this is more detailed than what we can just see here, and we need to understand each portion. So the regulatory portion is divided, just this portion we are able to see here is divided into the lac I, which codes for a protein called replacer, and then the operator, the promoter, and at the end of the Lac Operon we have got a terminator. So this which we can see here is called the terminator. That is the one which is going to end the process. Alright, so we need to understand this one in more details, just the regulatory portion. Let's go on and do that. And so we have got the three structure genes which we've already looked at: lac Y, lac Z, and lac A. So these are the structure genes on this DNA strand. So behind the structure genes, this is where we have got the regulatory portion. Now the regulatory portion is divided. We have got an operator just behind the structure gene. Apart from the operator, we have got the promoter, and then just much behind we have got lac I. Well, we need now to get into the discussion of understanding the functions of this. So the enzyme RNA polymerase attaches to the promoter, which is just behind the operator. This is where it attaches. That is where the enzyme RNA polymerase is going to attach before it can actually form gene product from these genes which we have here. And so if the promoter is the point of attachment of the RNA polymerase, so behind the operator, now the operator, what is the function of the operator? The operator, well, acts as a switch. This is what switches on and off. So if this is on, then RNA polymerase here is going to be able to move. So let's say that's RNA polymerase. So that means if it is reading on here, RNA polymerase will be able to move. If it is reading off, then there is nothing that is going to happen. So the RNA polymerase will just be on the promoter. And most of the times, it should be stated, most of the times the operator is in the off position or in the off mode. And why is that? That is because most of the times the bacteria is rather using glucose other than breaking down lactose, because lactose has to be broken and a lot of energy is needed, and so most of the times glucose is used instead of breaking down lactose. That is why the operator is mostly off. Okay, all right, so the question would be, what turns the operator on and off? Because we are saying it is either on or off, and when it is on, this is able to move the RNA polymerase. So there is this gene which you are able to see behind there, which is called lac I. So a gene known as lac I. Now I is standing for inducible. So this is lac inducible, and so just say lac I. So this one, which is behind the promoter, continuously synthesizes a protein known as repressor. So this one, time and again, it synthesizes a protein known as a repressor. Well, that is it. It is going to synthesize the repressor. Now, when the repressor is attached to the operator, so what happens is this repressor is synthesized, it is going to go and attach to the operator. And so when it is attached to the operator, this is known as the bond state, and the operator is off. And so this repressor which is produced is going to go and bind to the operator, and when it binds to there, then that would be off. Well, let's consider that. So we are saying Lac I is going to be able to produce the repressor. Now the repressor has got jaws which are able to hold to the operator, and most of the times actually the operator, this repressor is always okay, most of the times, because we are saying most of the times this operator is off. Because of that, it can only be off when the repressor is there, and so this should tell us that the repressor is most of the times attached on here. So this is what we are referring to as the repressor, which is produced by Lac I, or Lac inducible. Alright, so we are saying whenever the repressor is attached to the operator, this is called the bond state. They are held together, and the operator in this state, it is off. So in the bond state, the operator is off in that one. So therefore mRNA polymerase, because it is off, cannot move. Remember, we've got RNA polymerase at this point. It cannot be able to move going forward. So that is what happens. Well, now what is going to switch this on? That would be the question. And we need to get to know that 90% of the time this is in the off position. That means the repressor is always attached to the operator. Well, the repressor is normally held to the operator in the presence of glucose. So whenever you have got glucose in the body, the repressor is always there. Now, in the absence of glucose and in the presence of galactose, and that is, it should be absence of glucose and presence of lactose. These are the two conditions. These are the only two conditions which are responsible for the Lac Operon to work. So when glucose is there, and then even if lactose is there, the Lac Operon is going to be off. As long as glucose is there, the Lac Operon is always off. So when all the glucose has been used up, and now lactose is there, that is when this can be switched on. The operator can be switched on. Well, what happens then? So the repressor, we can see, has got what is called a lactose binding site. So the lactose binding site is the point where on the repressor where lactose is able to come and bind. So say this is lactose. That is the region where lactose is going to come and bind. So when this happens, whenever lactose binds to the repressor, these jaws are going to be loosed. They are going to be pushed like that. And so the repressor is going to be off from the operator. In other words, the repressor is going to be away from the operator. And because of that, when this is away, so the jaws have been loosed, and so the operator is now just free. Okay, the operator there is going to be free, and then this is going to just be like that. It's not held. So on the promoter then, where the RNA polymerase, the door has been opened for it to pass, and so this is what is called the on position. The operator is now on. Okay. So when the operator is free of the repressor, it is turned on and RNA polymerase moves forward on the DNA transcribing the gene. So it goes on and moves, thereby transcribing the gene of our interest. Now, the bacteria only produces the enzyme to digest lactose when lactose is available and not all the time. And that we need to get it well. It is not always, it is only when lactose is available and glucose is absent, not all the time. That is very important for us to understand. Okay. Now, another thing to get to know is behind the promoter region, just behind here, there is what is called a cap site. So let me just fit it there. So just behind here, there is what is called the cap site. Well, what does this mean? So cap is representing for catabolite, activator, protein. So that is the cap site, the catabolite activator protein. So this cap, the catabolite activator proteins, helps RNA polymerase to be held more effectively to the promoter. So just a little behind the promoter, the cap site, I mean not cap site, but the cap itself, not the site, but the catabolite activator protein helps the RNA polymerase here to be held more tightly to the promoter. Okay, because of this is going to increase the rate at which enzymes transcribe the gene of Lac Operon. So when we help the RNA polymerase to be held more tightly, that is going to increase the rate at which RNA polymerase is going to make gene products. Alright. Now, the cap protein is not always active. So this catabolite activator protein is not always active. Well, it is only active in a certain condition. So the cap protein is not always active. That's how it gets its name, because it is only activated by a certain condition. So that is, it is only activated during catabolism, and catabolism involves the breaking down, in this case, of lactose. So the only way the catabolite activator protein is going to be activated is when catabolism has to start to happen. That is the breaking down of our lactose. So that means it should be in the presence of lactose, in the absence of in the absence of glucose. That is very important. Okay. So if glucose and galactose are both present at the same time, that means this is a signal of catabolism to the cell. Okay. When these two, glucose and lactose, are present at the same time, so the bacteria is going to first use up all the glucose. Not until all the glucose has been used up, then we don't have any glucose. So the absence of glucose is what signals the catabolite activator protein to come in. So the absence of glucose is a signal for catabolism that lactose now has to be broken down. That is when the catabolite activator protein is activated. Very important to understand. And that is mediated by something. Well, for this to happen, for us to activate the catabolite activator protein, there's a help. A certain enzyme helps in the process, and that is called cyclic AMP. Now, cyclic AMP, this is cyclic adenosine monophosphate. So this is what helps, this is what mediates the process. Okay. So the signal that is sent. So whenever the glucose concentration in the cell is low, this is low, cyclic adenosine monophosphate, this we can see, we can also write it as AMP, cyclic adenosine monophosphate concentration is going to rise. All right, so this is going to rise. Now let's write that condition here, because without it we are going to error. So low glucose concentration low glucose concentration, this is going to mean CAMP concentration rising, and we are saying this is the cyclic adenosine monophosphate concentration rising. So this is important. So whenever you hear low glucose concentration, that means high CAMP concentration. Very, very important. Well, what now? What if the concentration of this one rises? What happens then? So when the concentration of CAMP rises, the raised concentration levels activate now what we are calling as the catabolite activator protein. So it is only the high concentration of this that activates CAP, the catabolite activator protein. So when this one has been activated, that is now going to be a signal that DNA glucose has been, so that is going to be a signal to DNA that all the glucose has been used up. That is a signal. So when this is activated, first, when we have got low concentration of glucose, there will be a high concentration of CAMP. This one, after being in high concentration, it sends a signal of activation of this, where whenever this one is activated, then the cell recognizes that, okay, glucose is done here. Well, when the glucose is done, then now we have to start breaking lactose. Okay, so that is how the process goes. Low concentration of glucose, high concentration of CAMP. This activates CAP. The activation of CAP is a signal to the cell that we need to form enzymes which should be able to break lactose. And so because of that, the cell is going to do what is necessary and allow lactose to come and bind here. When lactose binds on that site, then the jaws are going to open up. When the jaws open up, this is going to move up, and then RNA polymerase is able to move, and so you are going to form enzymes which are able to break the lactose, and so now you are going to have lactose broken down into glucose and galactose. So that is basically the function of the Lac Operon, and that is how the process goes. And we are going to have the glucose at the end, which is going to be used up now by the bacteria, and it will be also important for human being, because at this time bacteria is in the stomach. So if this is the bacteria in the stomach, then this process is taking place in the stomach. If you've not eaten anything that contains glucose, you've just drank milk, you'll not be able to suffer. And that makes the end of this video. Hope you enjoyed. Stay blessed.